Hey everyone, it's your host Russ Altman from The Future of Everything. Today we're rerunning an episode about Alzheimer's disease. It's a topic that touches many people. We still don't have a complete understanding of the disease, and that makes it harder to design effective therapies. In 2022, I sat down with mechanical engineer Ellen Kuhl, who offered a glimpse into the way she's using computational modeling to help improve our understanding of Alzheimer's. I hope you'll take another listen. Before we jump into this episode, I'd like to ask you to rate and review the podcast. It'll help others figure out if they're interested in the future of everything. Alzheimer's disease has impacted many people all over the globe. Almost everyone has a family member or a friend or knows somebody affected by this disease, which is characterized by a progressive dementia. Starts out with memory loss, but then it advances to other parts of the brains where you then get a profound disability and ultimately death. Alzheimer's disease is a priority for many countries as the population ages and we see it increasing in prevalence. We have very few drugs for Alzheimer's disease, although there are some recent ones that Ellen is very excited about, especially if we use them early in disease. Diagnosis is often too late, and so imaging methods are being developed that might be able to see the changes in the brain 10 or 15 years before clinical symptoms develop. Finally, we are trying to understand the physics of how the brain malfunctions because a deeper understanding of the detailed mechanisms of damage will help us fight it. Professor Ellen Kuhl is a professor of electrical engineering at Stanford University. Her lab specializes in using computational methods to understand living systems, including heart and brain. Ellen, you're trying to address one of the most important diseases in the world today, Alzheimer's disease. What drew you to this disease, and what can your research expertise offer to this very vexing problem?
Yeah, hi Russ, thanks for having me. So maybe a little bit of background. So my lab brings together biomedicine and physics and computer science. And what we try to understand is really how living systems evolve throughout the life cycle. And so a couple years ago, we were interested in got the idea of, wouldn't it be cool to study the human brain throughout the life cycle of the brain? And of course, the most interesting part of that is really how it degenerates as we age. And so this leads into what you just said, studying Alzheimer's disease with computational models.
And so what is the approach that you take? When you're using computers, do you do any kind of experiments on the brains, or do you rely on any data that people collect on the brains? And then how do you factor that into a computational model? And what are the kinds of things that these models can get us that other methods might not be able to get us?
That's a great question. So really, we focus mainly on data that's available. And you probably know that over the last, 5 to 10 years, there's been an explosion of people collecting data, making it publicly available. So for this in particular, there's a database that's called the ADNI database, where people collect data on Alzheimer's patients and healthy brains. And there's hundreds of data points. And what's cool is there's data points in time, so multiple studies over multiple years. And this plays into our hands because we're not only looking at the time snot picture of one patient at one point in time, but we really create dynamics models that can tell us how things evolve with the physics, really time evolution, how things evolve in time.
Yes. So tell me, let's step back a moment. How do you think about this disease? I mean, everybody knows that it's associated with dementia. Almost everybody has either a friend or a family member who's been affected by it. But when you look at this disease, what are the key features that draw your attention and that we need to understand?
Yeah, so that's a great point. So there's actually 4 what people call biomarkers. So these are the things that are associated with the disease. 2 of them are probably something you're very familiar with. This is proteins, misfolded proteins. So that's amyloid beta and tau. So these are these proteins in the brain that clot up and form clusters and clumps, and that then they can no longer function properly. And then in response to that, cells of the brain will die, the brain will shrink, and that causes actually the cognitive decline. And only then, actually, when we see the cognitive decline in people, we can diagnose it. So the real question or the challenge of the whole thing is that like years before we can see that someone loses memory or has cognitive decline. All this has already happened.
So tell me, do these proteins that you mentioned, do they have a normal function where they're normally doing something good and then something goes awry? And how early does the process start? How early could we possibly detect this? Because obviously, intuitively, you imagine that getting an early indication that somebody has the disease would give you more options for treatment.
Yeah, so for instance, these tau proteins, they stabilize the cells in the brain. So there's these nerve cells, these axons, the long cells that propagate all signals. And so the tau, if that actually clumps, can no longer stabilize the microtubules in these cells. And then everything breaks down, and the cells are no longer stable and in their position, and then they break down. So the two ends of the cells can no longer talk to each other.
I see.
So then actually that's a real indicator. This happens about 10, 15 years before it really builds up to a level that we lose really function.
And if we pay close attention, can we see these changes 15 or 20 years early, or is it really like sub-detectable? We can't detect it because it's too much of a subtle process?
So you can't detect it in a person in the behavior 15 years earlier.
Yeah.
And you could not, until a couple of years ago, you couldn't even detect it in a scan. So this is really, really new development. So the way it was detected under Alzheimer, when actually the person who developed this diagnosis for the disease after, which is now named, he actually cut dead brains. He sliced them, stained them, looked at a microscope, and saw these clumps. And of course, that requires a person is dead. And so now, only over the last couple of years, the technologies have been developed to neuroimage the brain with tracers for tau and amyloid beta. And you can, in the living brain, really look at they light up in the tracer. You can look at these proteins. And that's really, really cool because it's non-invasive. So you can do it anytime, as often as you want, get a real picture and the history of it. And that you can probably see, I would say, 5 years after it has started or so, when a little bit of buildup is there. And that's, of course, much, much better. But people wouldn't come naturally to get that scan, right? Because there's no symptoms.
You would have to have either some sort of screening program, and you would have to make sure that you weren't getting a lot of false diagnoses. It would have to be very accurate in order to justify doing it on the entire population. Otherwise, you get a lot of people nervous about a disease that they don't have. And okay, so that was a good background. So we know it has these clinical syndromes. You're telling us that there are these molecular changes that are happening 5, 10, 15 years early. The first good news that we've heard from you today is that there may be imaging modalities available now and in the future as they get better that will tell us about these changes. What are your computational models doing exactly, and what have you learned?
Yeah, so we bring together all this information, these different scans of these different proteins, of the size of the brain, the age of the person. So for instance, the brain shrinks 1% per year. Bad news, because your brain will be smaller than mine.
And this is normally even without Alzheimer's?
Yeah, in a healthy person. It declines at the biggest size at the age of 25, and then it starts to decline.
Oh my
and
goodness.
So you need to always take all these things into account when you can't just look at a single one brain and say, this is where it's at, right? And so you can do this with models. You can bring everything together. And what we do is we don't just do, say, a neural network or machine learning. We build in the physics. So we build in the whole history of things, how these proteins propagate across the brain. We use like partial differential equations, so propagation equations. We use kinetic equations that describe how they build up locally. And all this together brings all this information together and gives a much better picture than just looking at one single thing.
So that really does make sense. So is the model? So I guess my question is it that a few cells get sick, and do they transmit the illness in some way to the neighboring cells? And is that by actually giving these bad proteins to this nearby cells, or do they actually just send signals that mess up the nearby? Do we know how cell-to-cell spread of the disease happens within the brain?
That's a great point. We don't know exactly. So what people believe now is that it spreads like prion diseases. So these are diseases where the misfolded protein recruits healthy protein, and the healthy protein also misfolds on the misfolded protein. And then they break apart at some point. And so it's an exponential increase of this toxic misfolded proteins in the brain. And then, of course, we can model how this increase over time really affects the entire brain.
So forgive me this really detailed question, but I'm just trying to understand. Is it that the bad protein accumulates, and does the cell kind of burst open, and then those bad proteins get inside neighboring cells, or I'm just wondering how it spreads from cell to cell, and if do those proteins go from inside of one cell to inside of a cell next door? Maybe this is not known. I'm just trying to get a really clear
I think
picture.
that's the understanding for tau. So tau is inside the cell, amyloid beta is outside. So just spreads more like gradually across the entire brain. And so what's cool is because it's in the cell, you can also trace where it's going if you trace the connectome of the brain. So you travel within the axons. So that's something we can take into account in the model as well.
Yes, and I've looked at some of your papers, and it looks like you have these models that show that the spread is not just random, it's actually along the paths of the nerves. Yeah. And so does this give you insight as to like which parts of the brain might be affected early? Like I don't even know, are there certain parts where it tends to pop up and then spread? And what do we know about the spread of the disease in the brain?
Yeah, and that's actually interesting because it's not just spreading like to neighbors and then just like a wave. But it really travels along these axons, at least what people believe and what we can show with the model. So it starts in brain stem, and then it goes to the front of the brain, so frontal lobe, then the temporal lobe, which is top, the parietal lobe, which is the top, and then the occipital lobe. And then these regions all have a certain function, right? So you can actually trace the loss of function in Alzheimer's disease. So for instance, you have memory loss, or you have loss of face recognition, or loss of feelings and empathy. And so all this is in certain regions, and you can map out which regions lose function at which point.
Right. Okay. So that, I didn't know. So that's great. So it actually does have a standard way to progress, and that way actually is maps very nicely to the way that the brain is connected itself. Now we do also know, I think, that there are both environmental things that contribute to this. Like I think a history of trauma to the brain can sometimes bring it, and I think there's also like genetic predispositions. It kind of runs in the family. Are you able in your modeling to understand where those factors come in, or to model them to kind of personalize the understanding of Alzheimer's across different patients?
Yeah, we can totally do that. So we can see this disease in different locations. What you're saying, for instance, in football players, when they have chronic traumatic encephalopathy, there was also a seeding of these misfolded tau somewhere in the brain. And that might not exactly be like Alzheimer's, but Alzheimer's-like. And so you have the same kind of thing. So the model also works for that. And it also works for people who have genetic predisposition of this disease. And then it just happens earlier in life, right?
Yes.
But this pattern in the normal Alzheimer's, I think this spreading pattern is very, very stereotypical, very reproducible.
I'm struck that you're doing this really kind of forefront work, and you're by training you're a mechanical engineer. How do the doctors, the neurologists, the other people that I'm sure you collaborate, how do they accept somebody who's kind of not from within the group of like traditional Alzheimer's researchers? Have they welcomed you? Do they resist your models? What's the status of the kind of interdisciplinarity of the approaches to Alzheimer's these days?
Yeah, I think it's tough initially. I think you kind of have to be really hard looking for the people who really respect this. I've learned to kind of walk away from people who don't want to do it. But I found a lot of great people, and usually those are the people that really are looking at this as more the multidisciplinary approach. And then you can learn a lot of things from them. You can feed into the model what they know from mice models or from cell models in a dish. You can actually use this information and build that in like as rate constants in some of the very local parts of the model. So this is incredibly helpful to work with people of different levels and different background.
And when you make a prediction, so you run, one of the great things about models, computational models, is you can run them a zillion times, you can analyze them, and I'm sure at some point you make predictions. What are the kinds of ways to validate these predictions by working with experimentalists or neurologists? Have you made any surprising findings that then turned into a discovery because they then tested it and it turned out to be correct?
So right now we're testing it against this big database, so we can take out single one examples and test them. We can predict them. These things are developing as we speak, right? People are still screening and adding annual scans. And so we started this a couple of years ago, and we've made predictions to now, and we can go back now and compare against what people are measuring. So that's actually really exciting because it's really not known. And so we do predict something, and a year later we can go and see if that really happened.
Yeah, that's
and
exciting.
one really a cool example to play with is also there's been one drug that has been very controversial that's been just in the news a lot, And we can build the kinetics of this drug into the model, and then we can, for instance, test whether it's better to clear smaller clusters of these clumps of proteins or larger, or just not have them accumulate at all. So we would just play with these rate constants. Although we can't really exactly predict what the drug would do, we can play with what this would do or if this would be better and just compare different things. And so I think in that aspect, it's also very cool because it can accelerate drug development in general and explain what these drugs do.
Yeah, well, let me push you on that a little, because that has been, anybody who Googles Alzheimer drug will see a whole bunch of news. Are you finding anything that is either positive and reassuring and optimistic about the drug, or are there things you're seeing that are making you worry about it?
No, I think it's great. I mean, it's definitely a cool thing. People are mainly worried about the high cost of it.
Yes.
I think it's probably not going to treat people eradicate Alzheimer's in the people that are treating it with right now. But I think what's going to happen is we learn a whole lot of it, right? We can scan these people now. We can see really what it does. And the key, of course, as we said earlier, is to diagnose it super, super early so that this drug will be the more effective the earlier you start using it. But I think it's a great development because it gives a lot of insight, although it comes at a high price, of course.
You've done many, many studies about the spread of COVID in the last couple of years. Where did your interest in COVID arise, and how did you make this apparent lateral move from Alzheimer's disease to COVID?
So first of all, of course, we're all interested in it. Many of us are probably tired of it, but I'm going to tell you why I'm still excited about many of the things I've learned. So we've actually worked on the brain, and we talked about that we have developed a model, and this model for the brain had certain nodes. There was the regions of the brain, and that a tau protein would spread across the brain along these highways, the axons. So now we used exactly that model when COVID came. We thought, hey, can't we just recycle this and use it for COVID? And so the brain regions were the cities, and the axons were the plane connections between them. So
you didn't have to do a big
No, not at all. So the whole infrastructure was there. The only thing that we changed, obviously, is the reaction term, which is locally how the tau increases at a position, and now it's how COVID would spread in a population in a city. But the mixing of populations was exactly like traveling in the brain. So it was traveling through the air traffic control network. And so we got that network from air traffic. And then we used it a lot at the beginning of COVID, because remember, there was all this discussion about should we open, should we shut down? Especially Europe closed all their borders very, very early.
Yes.
And so they asked, actually, can you model this? So we predicted a couple of things, like what would happen when they open, when should they reopen, when should they close? And so they was actually really using the same model. And then that led to a class. So just out of interest, we're like, oh, why don't we develop a class? And then we had a lot of students, and they were doing class projects. And then we did studies of campus reopening with this model. So the students themselves studied how and when it would be safe to reopen campus. Because you probably remember that in the early summer of 2020, there was all this campus super spreading stuff. And so, that was then student projects. So they loved it. They did all these studies, wrote papers actually about it.
Yeah, so before I ask you about the results and what you learned, I just want to this is an amazing example of why, you never know how research is going to impact the world, because here you are working on Alzheimer's disease, and I just am really struck by the fact that your model was a perfect match to the spread of COVID, with just a little bit of cleverness, and of course a lot of cleverness on your part. But it's a great lesson for why you can't always tell how your efforts are going to lead to fruit. So having said my little speech, thank you for listening. You know it very well, but I really want to highlight it. What did you learn about COVID? So what do the models tell you? And especially things that are surprising, and maybe with the benefit of hindsight, is it clear that there's things we should have done differently?
So we looked, for instance, very early about asymptomatic transmission. That's pretty easy to build in the model because there's just a different part of transmitting the disease. And it was surprising that people didn't accept that. There was a long discussion always when people were saying, and even the CDC was going back, there is no asymptomatic transmission. And then people were like, how does it then transmit? And so there were all these discussions. And I think the models we have tested that pretty early. And we're like, there must be some transmission that we don't record and see, because otherwise, how is this going to even spread? And then...
so you couldn't even explain, you were observing data that didn't make any sense unless you included... Yeah. I see. So it was almost like a proof that something was missing because the models just weren't adding up.
It's kind of funny because, I mean, when you look at the data and you have a model in mind and you do this a lot, then you kind of see, hey, there must be some term to explain this. It's not there.
Right.
And so that was kind of intuitive. And some other things were also pretty intuitive pretty early. So this is all these discussions about closing places and opening places. We worked actually with the government of Newfoundland, which is a small Canadian province. It's an island, and they were totally isolating themselves. And then they were sued, and they're like, can you help us to figure out how we open up our airport? And so we worked with them directly. And it's kind of interesting because you're like, yeah, this is so clear. And in a way, exactly what the model predicted happened. But I mean, of course, a lot of things didn't happen. So I think the real problem with COVID is that a lot of people used models that have never used models before. And then they came out with predictions where case numbers were negative or were larger than the world population within a week. And so I think that people lost trust in models quite a bit during COVID. And you probably have experienced it too. And that was really a disgrace to modeling in a way.
Yeah, so, yes, and that's really interesting that you said that some people just kind of, like, it sounds like you guys used your models in what I might call a responsible way. By the way, even using it in the class is a way to test it, because then you have a bunch of smart young people using it, and they will, you and I know, they will definitely tell us if they think that our models are stupid or wrong, and they're not very shy about this. So that's also a very interesting point, and I know you wrote a book about this. So is that where the book came from? Was your kind of seeing the misapplication of mathematics to the COVID epidemic?
Yeah, totally. So I was very disappointed in a way that at first everybody's like, oh, modeling, yeah. We will know in a month from now where everything goes. And in 2 months and 3 months, this will happen. And the next person walked out and like, no, this is going to happen. And so I think, people who take modeling seriously kind of knew that this was never really going to be predictive for more than 2 or 3 weeks out. And so we were like, okay, we have the responsibility to educate people. So I gave a lot of talks during the time online, and just really to teach people not to blindly use models and also not to lose trust in the models, right? Because our community was like feeling very offended by people saying models are useless. Right. We can just look at the data.
So let me ask you about that, because for people who are not experts and who are just listening to the news, trying to make best decisions, for their kids in school or for their business, are there things, so separate from trying to get your modeling colleagues to do a better job, is there anything for the public, is there anything that the public should be asking when they hear about a model so they can kind of check it and make sure that it's reliable? Like, what would you tell somebody who's not an expert and who's confused about all of this?
It's funny that you say that, because we have taught ourselves during COVID actually came up, used a lot of machine learning and Bayesian methods, it's called. So it has credible intervals. So it tells you where your results are within all possible results. And the narrower these credible intervals, the better the model. And so, of course, most models don't report that. But sometimes it's now even reported in the weather forecast. And that gives you kind of an idea how confident you are in what you're predicting.
It's like the plus minus.
Exactly. And and sometimes it's like spanning the entire graph, right? And then you kind of know it's a little bit useless. But and that's actually, we learned that too. We never really looked at that much before. But now we're like, everything we do, we want to have a very narrow credible interval. And if you look at some models that came out of the White House, they had credible intervals after 2 days. They just blew up, right? So.
Oh, that's interesting. So a model may be fine for one period of time, like the next week or two, because the plus minus is very narrow. But then you have to look carefully, because at 2 weeks or 5 weeks, it might be not credible anymore. So it's not that the model is always good, always bad, but you have to kind of look at where it's performing well. This is good. I want to ask, I don't know, but are there other things that a layperson should be cautious about when they hear about a model?
Yeah, I think this is the best way to test it. I wouldn't really, I guess the more data goes in the model, the better in general. In COVID, we had a lot of data, right? And so I think that was a good thing. For us, actually, it was a good thing to learn about machine learning, because we had all of a sudden, everything was documented, was available, was super well cleaned up and available to everybody. So that was actually, I think one thing we could learn is that this was very helpful for science. Everything was accelerated, right? The developments, the drug developments, everything. And that was just really because data was shared so openly and publicly and clean.
So the sharing of data might be hopefully one of the positive legacies of COVID, at least when there's an emergency and people let down their guard, because people who don't do this for a living might not know, but people often hold on to their data because they think it's valuable and they don't want to share. But in many cases during COVID, there was this sense that, well, we have to share because people are dying and this is an emergency. And it really did lead to accelerated progress in many areas. What about the interpretability of a model? Like, is there a difference between a model where you can explain exactly why the model came up to a result versus, well, it's a very good model, but it's too complicated and I can't really tell you why this is the answer?
So I have actually stepped away from using too complicated models. I think when you're only modeling person, you add terms to it, make it more complicated and stuff. And at the end of the day, you have so much screws to tune the model to something that you don't really see any correlation to the data. And I think what we've done over the years is really reduce the complexity of the models. Really, with every term that we use, we understand. And I think that's pretty valuable because everybody can make it more complex and can fit a curve. I mean, the more degrees of freedom, the more complex the model, the more easier it is to fit. You totally overfit what you're seeing.
Yes.
And so this is a good message to just reduce it really to the core, what you need to do.
You have been listening to the Future of Everything podcast with Russ Altman. I want to remind you that the Future of Everything started out as a radio show on SiriusXM, so you'll hear references to that. Now it is 100% podcast. We have more than 200 interviews in our archive, and they cover an extraordinary range of topics. If you're enjoying the podcast, please consider subscribing or following so that you can be alerted to every new episode and never be surprised by the future. Maybe tell your friends about it too. Definitely consider rating and reviewing it. That helps us grow, improve, and also spreads the word. You can connect with me on Twitter at RBAltman and with Stanford Engineering at StanfordENG.